Hi everybody, welcome back to Pathology 101. Today we're going to speak about cell injury and how the cell adapts to stress. Let's get to it. So normally, cells maintain a steady physiological state, and that's via homeostasis, right? So when cells are physiologically or pathologically stimulated, they can achieve a new steady state by undergoing adaptation. And that makes sense. When the environment of the cell changes, the cell is going to try to adapt to that new environment, correct? And it does so by adapting. So any change in stress can result in cellular adaptation, whether that stress has increased, decreased, or just changed in type. That's going to change the type of adaptation that takes place. So cell injury is going to happen if stress overwhelms the cell's adaptive capability. And let's talk more about cell injury. So a normal cell in a steady state would undergo adaptation when its environment changes. And that's a new steady state that's going to be achieved via adaptation. If the stress or the change is too much for the cell to handle, that cell is going to be injured. And there's another scenario for the cell to get injured as well. If the stress was so severe that it was inherently harmful, it's going to injure the cell directly. And cell injury is subdivided into reversible and irreversible cell injury. Reversible cell injury, of course, can go back to being a normal cell in a steady state because it's reversible, right? And irreversible cell injury will lead to cell death, which is of two types, and that is necrosis and apoptosis, all right? We're going to speak about them later. Let's go back to cellular adaptations. We're going to speak about hyperplasia, hypertrophy, atrophy, and metaplasia, all right? And I have to say, for the sake of the board exams, it's not sufficient to just know what each one means. You have to know the mechanism with which they take place as well. So let's start off with hyperplasia and hypertrophy. An increase in organ size is going to happen via increase in cell number, cell size, or both. Hypertrophy, which is an increase in cell size, is going to take place via increase in the size of the cytoskeleton, all right, and which is made up of proteins. So I'm going to do that by gene activation and protein synthesis, together with the production of organelles. So I'm going to sort of increase the size of the covering of the cell and the contents of the cell, all right? So hyperplasia, an increase in the number of cells, is going to take place via production of more cells from stem cells. And what's important is that these two processes usually take place together. An exception would be permanent tissues, and we're going to speak about them in a bit. So hyperplasia and hypertrophy can be physiologic or pathologic, very important. And physiologic hyperplasia is further subdivided into hormonal and reactive hyperplasia. So let's take an example for each one. An example for hormonal hyperplasia would be the enlargement of the breast during puberty. And an example of reactive hyperplasia or compensatory hyperplasia would be the enlargement of a liver in an organ donor and after taking a part of that liver for donation, all right? So you could find the liver has increased in size because it produced more cells. So pathologic hyperplasia, that's really important, can lead to dysplasia and carcinoma. We're going to speak about dysplasia later and carcinoma in a different video, but I want you to set this aside and sort of set the exception of benign prostatic hyperplasia. This is a very important exception to the rule. Benign prostatic hyperplasia is a type of pathologic hyperplasia that does not lead to dysplasia or cancer. Again, BPH does not lead to dysplasia or cancer. So let's branch off to the types of tissue in the body. We have permanent tissues, and that includes cardiac muscles and skeletal muscles and nerves. And what's important is that permanent tissues do not have stem cells, and therefore they do not undergo hyperplasia. They can only undergo hypertrophy. Let's contrast that to bone marrow stem cells, which are always undergoing hyperplasia, correct? Always producing new cells, correct? And stable tissues. Stable tissues include the liver, the kidney, and the pancreas. And stable tissues are sort of quiescent. They're sort of in sleep mode. And whenever you turn them on, they can produce more cells. So labile tissues include the hematopoietic stem cells, and these are cells that are always producing more cells, all right? And another example would be the lining of the intestines, for example. Okay, so let's go to atrophy. And decreased size and number of cells, that leads to decreased organ size. If I'm going to decrease the organ size, I'm going to decrease the size and number of cells, and that makes sense. It occurs due to decreased stress. Alright, so decreased stress, decreased organ size, decreased size and number of cells. Alright, so what's an example of decreased stress? Muscle misuse, denervation, diminished blood supply, poor nutrition, many examples like senile atrophy, for example. You don't have to memorize them, you just have to know the concept. So it's decreased stress. An example would be somebody who stops going to the gym and then, muscles shrink because of misuse, alright? Decreased cell number takes place via apoptosis. And you could think of apoptosis as programmed cell death or cellular suicide, alright? Decreased cell size, on the other hand, is going to happen via decreased protein synthesis or increased protein degradation, or both. So, how does protein degradation take place? This is going to happen via two mechanisms as well. So, the first one is autophagy, and that's when the cellular components are sort of placed into vacuoles and destroyed within the cell. And the ubiquitin proteasome degradation pathway, which is going to handle the cytoskeleton, so the protein. So ubiquitin is going to tag the protein, and the proteasome machinery is going to destroy the protein, the tagged protein. Alright? So let's move on to metaplasia. So we've discussed that decreased stress leads to atrophy. A change in the stress type is going to lead to a change in the cell type, and that's metaplasia. And this mostly involves surface epithelium. That's really important. We're going to take a couple of examples for that as well. And what is the point of metaplasia? The point is to produce a new cell type that is more tolerant of this new stress. Change cell type, more tolerant. A very important example is Barrett's esophagus, where the lower esophageal epithelium is going to change. Okay, so this is the esophagus and it goes down to the stomach. So the lower part right here is non-keratinized squamous epithelium, and that makes sense. It wants to handle that food bolus and the friction that it's going to create, alright? And so during gastroesophageal reflux disease, acid is going to reflux up, and the esophagus is going to try to become more tolerant of this acid, so it's going to change into the same epithelium that protects the stomach, which is non-ciliated mucinous columnar epithelium, and that's really important. You need to know all of this. Alright, so what the esophagus was, what happened, and how it's going to better handle the acid. Alright, so other examples include keratomalacia. Let's talk about that for a minute. So we all know that vitamin A deficiency leads to night blindness. Let's take that one step further and say that vitamin A is also important for the differentiation of epithelial tissue. And when you have a deficiency of vitamin A, the conjunctiva of the eye is going to have an issue in differentiation of its epithelium. It's going to scar, it's going to become really sort of tough and dry, and that's keratomalacia. Another example would be myositis ossificans, where the bone forms within the muscle. And why would that happen? It could be due to trauma, for example, and then you would find bone forming in the muscle. Alright, so Barrett's esophagus, very high yield. Stem cells undergo reprogramming, resulting in metaplasia. So this is the mechanism with which metaplasia occurs, and that's really important. Alright, we've discussed every mechanism so far. Metaplasia is reversible. That is really important. It's really important. How is it reversible? You should remove stress. So metaplasia can lead to dysplasia and cancer, very important as well. However, a very important exception is apocrine metaplasia. What is apocrine metaplasia? It is a disorder that takes place in the group that is fibrocystic changes of the breast, alright? And apocrine metaplasia is the exception to the rule. So apocrine metaplasia will not lead to cancer. It will not lead to dysplasia or cancer. Again, metaplasia is reversible, just remove the stress. However, it can lead to dysplasia and cancer. The exception is apocrine metaplasia. Let's move on to dysplasia. Dysplasia is described as disordered cell growth, alright? It often refers to the proliferation of precancerous cell. Usually results from long-standing pathologic hyperplasia or metaplasia. We've already said this. Pathologic hyperplasia can lead to dysplasia in cancer, metaplasia can lead to dysplasia in cancer, right? So dysplasia is reversible, very, very important. With cessation of the causative stress, dysplasia is reversible. However, prolonged exposure can lead to carcinoma, which is irreversible, and this is a very important concept. So dysplasia, removes stress, goes back. Dysplasia, stress continues, moves forward. Carcinoma, and that is irreversible, and that is the very important difference between them. I'd like to thank you for tuning in. Please follow, subscribe, and like the video. And if you have any questions, you can email me. Thank you.